We are also now partners with UIS for celebrating the International Year of Caves and Casts, which is going to be ending in December this year. This program is part of the celebration. Then we have also recently launched our blog, which is basically an interdisciplinary platform for sharing your experiences, any updates, or any information that you might feel like sharing or writing about regarding casts and caves and any subterranean habitat. You can write to us on our email ID, which is on the screen right now, and we will also share it again in the chat box. Anybody is welcome to contribute to the blog. Coming back to the topic of discussion today. So Dr. Mahi is a research fellow at the Birbal Sahni Institute of Paleosciences in Lucknow. She has been researching life histories of plants and investigating the evolutionary and migratory history of plants, which sounds like very interesting, and I'm sure you're all very eager to find out what exactly is it that she's working on. So basically recovering past data about plant life, about what our planet was like from a biological and a geological point of view, based on the deposits that are recovered from the Earth's surface. So subterranean um, fossils, basically. So with that, I ask Mahi to take it forward and please start sharing his screen and we can
Yeah,
begin.
thank you, Amrita, for that introduction, and thanks to the team for inviting me as a speaker in this webinar. I'll just share my screen. Is it all right? Can everyone see
Yeah,
my
it's okay.
screen? Yeah, all right. So, a very good evening to everyone. Let me start by introducing myself once again. I am Mahi Bansal, and I have recently received my PhD on paleosciences. And as you just heard, this word paleosciences contain a prefix paleo, which means past. So, I basically research about the life of plants in the past, and for that, we dig up remnants of plants from beneath the Earth's surface. And today I'm going to tell you about some of my experiences on how do we recover fossil pollen from mines and subsequently use them to reconstruct paleo vegetation and paleoclimate as well. So first of all, let us have a look at the picture of a mine. So these two pictures are from Vastan lignite mine, which is situated in Gujarat. So a mine is nothing but an excavation in the earth surface from where lignite is extracted. And lignite is a low quality form of coal that we generally use in electric power plants and other sort of industries. So now imagine this barren lands to be once covered with tropical rainforest that support the lives of several plants and animals. A little hard to imagine, right? But around 55 million years ago, India was situated at the equator when it was indeed covered with these forests. Uh, and when not just at the Indian subcontinental level, when we see at the global level, this is the present distribution of tropical rainforest. But around 50 to 60 million years ago, these tropical rainforests were expanded up to higher latitudes. Now a question arises, then why did these rainforests restrict to certain areas? So to understand that, we need to have an understanding of the paleogeographic history of India, which is, or in other words, we can say we need to understand how the Indian plate changed its or shifted its position throughout the time. So around 180 million years ago, the continental configuration of the configuration of the continents we see today on the globe was not the same. So around 180 million years ago, when the dinosaurs were still alive, the continents of Africa, South America, Australia, Antarctica, and India all were combined together as a single landmass. And then gradually they started to split apart around 110 million years ago. So this is the configuration the continents attained. And here we see that the Indian plate is situated in the southern hemisphere and was still attached to Madagascar. So this red part here that is attached to the western coast of India is Madagascar. Then, around 67 million years ago, when the dinosaurs were getting extinct, India had detached itself from Madagascar, but it was getting closer to Africa again. So, one interesting point here to note is of all the continents, India was the only landmass that traveled farthest, that too in the northward direction. And as it continued its northward movement, around 56 million years ago, it reached at the equator, and around 10 to 15 million years later, it collided with the Southeast Asia. And this is pretty much similar to the present continental configuration. So this is about the travel history of India through deep geological time. Now, imagine this landmass as consisting of two different continents. When these continents started to split away from each other or to depart from each other, a rift is created, and with time, this rift gets deepened and eventually filled with water. Now, with the ongoing displacement, the two continents are completely separated from each other. Likewise, when Indian plate separated away from Madagascar, it got accommodated at the equator, and we all know that at equator, the solar heat maximum, there is a lot of solar heat received by the equator. So when the Indian plate was at the equator, it received a lot of solar heat that caused the water to evaporate and ultimately led to the onset of precipitation. And the amount of rainfall received by the Indian plate led to the emergence of tropical vegetation. Now, with time, this tropical vegetation is decayed and gets trapped under rock and soil deposition. So, the overlying rock and soil deposition uh, creates a lot of heat and pressure that converts the organic parts of the plant into fossils. So, the into lignites actually that contains fossils. So, the land which was once covered with tropical rainforest is now enveloped with lignite bands, and these lignite uh, represent the remnants of forest, as in the form of fossils. So, among other parts of the plants like bark, stems, spores, leaves, we also find well preserved pollen fossils in lignites. This is a graphical representation of a pollen grain. We all have studied the process of pollination in our primary classes, where a pollen is transferred from anther of a flower to the stigma of the same flower, of the same flower, or the same flower of another plant, and to enable the process of fertilization. So these pollen are very tiny in size and pale yellow in color, and these are the main reproductive part of a flower. The outer wall of this pollen is made up of a compound called sporopollenin, which is highly resistant to chemical and physical weathering, and that is why, despite lots of heat and pressure exerted by the overlying rock and soil deposition, we get well preserved fossil pollen in lignites. So these are some real pollen images that belong to different plant families. And these yellowish kind of images were taken through this light microscope, whereas these black and white guys were taken through this scanning electron microscope. These are some more pollen images here. These two pollen images belong to a plant family called Ebonaceae, and these two are from the Linaceae, which is commonly known as linen. So when we look at these pollen, we see that they are ornamented with very beautiful ornamentation or very beautiful sculptures. These sculptures are one of the identifying characters of fossil pollen grains. For example, this rugulate type of structure is exclusive to the pollen of Ebonaceae family, and we do not find this kind of structure in any other plant taxa. Similarly, these round protrusions, which we call as gemma or clava, are characteristic feature of Linaceae pollen. So, in other words, we can say that a plant family is distinguished by a set of pollen characters. So coming back to our question, that how do we extract them from sediments that are deposited beneath the Earth's surface? So to do that, we first conduct field work. This is a picture of a lignite taken from quite a distance. And when we move closer to this mine, we see well differentiated sedimentary layers here. These sedimentary layers are being represented with these vertical red and black lines. And each band here represent a different kind of lithology, meaning like some band are consisted of maybe sand and some other maybe consisted of clay or silt. And these black bands here are of lignites. This is just to show you how these different lithological bands are stacked up uh, in the nature. Okay, so we go to the field, we measure each sedimentary layer or each sedimentary band and create a litholog. So with this litholog, we have an overview of the whole mine. For example, we know that at the basalmost part of the lignite of this mine is made up of sand, and then we have 5 meters of lignite, followed by 7 meters of mud, clay, silt, and so forth. Here we are just discussing from where to take the samples, and in the rest of the 3 photos, this person is collecting samples for us. Then we just pack these samples like this and label them. Here, this poor chap sitting in isolation is me jotting down everything, every little detail of the field, like the number of samples or the nature of samples, whether they are sand or clay, or the height from where the height of the strata with respect to the base of the mine from where the sample has been taken. And this is just me telling you all that not to worry about your skin when you are working in a mine. And here, I guess I just decided to put on some lignite powder instead of conventional talcum powder. So anyway, these are just few perks of working in a mine. And in addition to work, field exploration is fun too. So here is my team relaxing a bit after completing their sampling work, and the joy of having food after doing the extensive field work is clearly visible on their faces here. All right, so coming back to some more geological structures, this single chunk of rock here is composed of several laminations. And trust me when I say this, each lamination must have taken thousands of years to get deposited. And when these horizontal layers gets curved because of the undergoing displacement of Earth, they create a fold. For example, this brown layer here was almost flat once, but when the compressional forces were applied from the opposing ends, it got curved and created a fold. And this is what happens when we geologists go crazy in the field. So we do not work on mega fossils basically, but while strolling through the field, we just came across this beautiful expression of leaf fossil. So we just decided to look up for more fossils. And then we collected it and donated them to the mega fossil lab in our institute. And not just the part of the plants, these lignite mines also contain some creatures from the animal kingdom. So this fossil is of bivalve, a marine invertebrate, and this is some crawling creature. And don't expect me to provide its name because I do not know what it is. So yeah, that's how our tiring but enjoyable trip ended. So coming to the lab work, so 10 grams of each sediment sample was crushed and put in these jars, and then these samples were treated chemically to remove any unwanted particles from them, like silicates or carbonates or any other thing. So then these solutions were washed and sieved to get a pollen concentrate in these dishes. So we pan these dishes, and then a very fine fraction of pollen concentrate was taken out with the help of this pipette and spread through the glass slides. These glass slides were then put in the oven until they dry completely. So once dried, the prepared palynological slides were seen under this light microscope and scanning electron microscope. So with the help of scanning electron microscope, we are just able to see the exine ornamentation or the ornamentation on the outer wall of the pollen more clearly. So these small entities are the different types of pollen, and this is the enlarged image of a pollen grain. Now we compare the pollen morphology of fossil with the pollen of living plants that are already published in literature, and we also extract some fresh pollen from fresh buds. So the next few slides are about the comparison between the fossil and living pollen grains. So this is a fossil image of a pollen grain, and this is the enlarged image of a pollen that belong to the plant Diospyros. See the striking similarity between the two images. Then we have the fossil and living pollen of linum. Then these are the fossil and living pollen of plant bombyx. And this is Paraschoria. I was so awestruck to see the striking similarity between the two grains, of which one is around 55 million years old and one we find in the present time. And similarly, we have fossil of Shorea robusta, which is commonly called as sal, and this plant is now exclusively found in the central India. All right, so when I started this talk, I mentioned about this mine, and I also mentioned that this mine was once covered with forests. So how do we know that? For that, we conducted a field survey in the Vastan lignite mine. We collected some samples there, and we processed them to extract pollen. So these are the pollen fossils recovered from the Vastan lignite mine. This column here lists down all the fossils that were recovered, and these are their living relatives. For example, we recovered a pollen fossil named as Phobia tricolpitis alveolatus, and this pollen shows very much resemblance with the living Dipterocarpus plant. So similarly, we recovered around 27 fossil types from Vastan lignite mine that show resemblance with the pollen of the plants that we currently find in Western Ghats. This is an easy representation of the distribution of rainfall across the Indian subcontinent. And this blue highlighted area is the Western Ghats. And as we all know, Western Ghats are very well known for um, their ability to support the survival of plants and animals, and they are among the topmost biodiversity hotspots. So the presence of the fossils of these plants and animals in Vastan lignite mine suggests that the forest of Western Ghats were once expanded up to Gujarat. And similar to Vastan lignite mine, we have other lignite and coal deposits spread across the India, which are represented here with these black blotches. So imagine there was a time when most of the India was covered with tropical rainforest, but with the changing climatic conditions, the amount of rainfall started to decline and the number of dry seasons started to increase across much of the Indian states. And as a result, the tropical rainforest retracted themselves to this patch along the Indian western margin. And so we also conducted similar kind of study in Jabalpur. Jabalpur currently is situated around 23° north, and but around 68 million years ago, it was situated below equator in the southern hemisphere. So again, we collected some samples from the Jabalpur and then processed the samples for palynological study. And this is the pollen diversity we retrieved from the central India. We also recovered some fossil pollen that shows their affinity with the plants that are not even found in India in the present day. For example, this Kilmaria pollenitis eosenicus, this is a pollen fossil that shows its affinity with Sarcolinaceae. And this particular plant family is currently endemic to Madagascar. Similarly, we have received a pollen fossil that is comparable to the genus Tetrachedium, which is now restricted to Africa. Now, there is a widely accepted assumption that these fossils must have been existing in the similar ecological and climatic conditions in which their living counterparts are existing today. So following this assumption, we made a table that lists down all the fossil taxa we recovered from the central India. This is just a representative table, so I haven't listed down all the fossils here, obviously. And then we wrote down all the nearest living relative against the fossil taxa, then the habit of the nearest living relative, whether it is a tree or a fern or shrub or whatever. And then we jotted down the habitat of the living relative, whether it grows well in sandy soils or it is grows well along the margin of the coastal area or what. So based on this assumption, we assigned the vegetative association to the fossil pollen, like to see whether these fossil pollen belong to the coastal fringes or they are a part of the tropical forest flora, or they grow as forest outliers, or they are the part of upland flora. So this is the final reconstruction of Central Indian vegetation, where we observe that a lot of mangroves and coastal flora grow along the water body margin. And then as we move inwards toward the terrestrial side, we get various forms of tropical rainforest, followed by the pollen fossils that belong to the plants that are forest outliers, and then followed by the upland flora. Now, when we derived this uh, vegetative reconstruction, my team was intrigued by a point that was there really a water body resource that facilitated the growth of these mangroves and coastal flora within central India? So the answer is yes. Another group of pollen workers have suggested the existence of these marine seaways within central India that might have enabled the sustenance of this coastal flora within central India. So after reconstructing the paleovegetation, we decided to reconstruct paleoclimate as well. And to do that, we followed the coexistence analysis. To understand this, let us try to read this graph here. So let us assume that we recovered a pollen assemblage from a region consisting of fossil taxa A and B, and this fossil A belongs to plant A dash, and this fossil B belongs to plant B dash. Now we will plot this the temperature ranges for A dash and B dash, and from this graph we see that the temperature values for plant A dash ranges from 4 degrees Celsius to 12 degrees Celsius, and for plant B dash the temperature values ranges from 8 degrees Celsius to 16 degrees Celsius. So now the overlapping values here shown by this blue shaded area tell us the temperature range in which fossils A and B were coexisting in prehistoric times. I hope I am making some sense here. So following this concept, we plotted the temperature and precipitation values for the nearest living relatives of all the fossil flora we recovered from the central India. And the results revealed that the central Indian vegetation was consisted of tropical rainforest flora that was existing under the climatic conditions of mean annual temperature around 24.5 degrees Celsius and mean annual precipitation of 1,890 millimeter. Moving on from here, I would just like to briefly talk about the migratory history of plant family Dipterocarpaceae. So you all might have heard about the sal wood that we also use in the reconstruction of our houses. So this sal wood is commonly found in central India, where people use it as their source of survival, also economic survival. So this plant family Dipterocarpaceae is further subdivided into 3 subfamilies: Pacaramoideae, which is endemic to South America; Monotoideae, which is restricted to Africa and Madagascar; and then Dipterocarpoideae, which is restricted to South and Southeast Asia. So we have recovered fossil pollen of these fossil pollen of Dipterocarpaceae fossils belonging to its different genera: Dipterocarpus, Dryabalenops, Shoria, Parashoria, Vateriopsis, and Monotus from Indian sediments. And in addition to the Indian sediments, we have also received Dipterocarpus type of fossil from the African Maastrichtian sediments. Maastrichtian as in 68 million years old sediments. So one interesting point here is these Dipterocarpus, Dryabalenops, Shoria, and Parashoria are still, we still find them in Indian or other Southeast Asian regions. But Vateriopsis is currently endemic to Seychelles, and Monotus is currently endemic to Africa, but we recovered their fossils in the Indian sediments. So these are the pollen fossils of Dipterocarpus type. Dipterocarpus type. Then these 3 images are Dryabalenops type pollen fossils. Then we have Vatica. Then Monotus type pollen fossils. Then we have Vateriopsis. Then we have Parashoria type of pollen fossils, Shoria albida, and Shoria robusta. So I'll not go into the detailed methodology here, but we just need to know that based on this pollen information and the DNA information of Dipterocarpaceae, we generated a phylogenetic tree. So we all might have created a family tree for ourselves when we were in primary classes, where we depicted the family relationship in a tree-like structure, where we started from the higher generations of our grandparents at the top. Then coming down, we used to show our parents and then uncle and aunt, and then we used to show how we are related to our cousins. So similarly, the phylogenetic tree for Dipterocarpaceae do tell us that from where did this family originate, when did this originate, and how did this family migrate from one place to another place. So the results revealed that Dipterocarpaceae originated around 102 million years ago in Africa. So this was the time when the Indian plate was situated too far south in the southern hemisphere, and it was under arid climatic conditions that could not support the growth of moisture-loving Dipterocarpaceae family. But as the Indian plate moved towards northward, it made a contact with northeastern Africa through a series of island arcs, and this facilitated the dispersal of Dipterocarpaceae forms from Africa to India. And the establishment of this corridor is also corroborated by the presence of similar type of Dipterocarpus fossils from northeast India and northwest India. Moving further in time, when India collided with Southeast Asia around 45 million years ago, it transferred all of the Dipterocarpaceae forms from India to Southeast Asia, and that's how in the Indian plate acted as a carrier in transferring the Dipterocarpaceae from Africa to Southeast Asia. I guess that's all from my side for today. Thank you.
Yeah, thank you. It was great.
Thank you so much, Mahi, and everyone else for attending. If there are any questions, I request you all to put them in the chat box, or you could just unmute yourself and ask. I think Disha Gupta has a question about how
Yes.
you identify the pollen not found in India.
For that, we refer various pollen catalogs that are established or that are published already from different continents.
Okay. I mean, my concern is like there are so many. So, what is the level of accuracy? So that is like really very strenuous on your part.
It is actually. We tabulate a lot of pollen characters. Like it sometimes goes up to 100 pollen characters, and then we try to match them that to which plant family our pollen best matches with.
For example, like how much dedication of how many hours does it take to find just for a single, identify just a single pollen?
Uh, I should say it does not take hours but months to actually confirm the identification of fossil pollen with their living counterparts. Yeah, we
Okay,
have to refer a lot of literature.
just for
So,
one pollen, 3 4 months, right?
yeah, not just for one pollen. Actually, we when we scan the slides, we retrieve a lot of pollen fossils, and then we have to keep the morphology of these pollen fossils in our mind, and then we refer to the literature, and that's how we identify several pollen grains at the same time.
Okay, so that's your experience that helps you.
Yeah.
Okay.
Hi Mahi. I
Hi.
have a question.
Yeah.
So I was just curious. So it's been so many millions of years, but some of the pollens have not changed. So have they not undergone any genetic, like change? Any
Uh,
mutations?
yeah, actually, very nice question, Shruma. Here, when we compare the fossil and the living pollen, no, so the main change has occurred in pollen size. So the fossil pollen are much larger than the living pollen. So this is the major change we have, yeah, perceived so far.
Right, but not like genetic change.
Oh, no. So there are some regulatory genes actually. Uh,
Mm.
so these regulatory genes do, like when a fossil pollen or when a plant is adapted to a new ecological conditions, no. So these regulatory genes modulate the internal functioning of the plants to get adapted to the new ecological conditions. So
Okay.
it is not the mutation and all. It just modifies. Or it just made the plant capable of living in the new
Adapted.
ecological conditions. Yeah.
Right.
Yeah.
Thank you. Very
So,
interesting. Mm.
yeah. May I ask a question?
Yeah, sure.
Yeah. So when you are retrieving the pollen from, say, the sediments, right?
Yeah.
So what is the like my question is regarding the viability of that pollen regarding its main function. Does that is that retained?
I'm sorry, I guess I didn't get your question. Can you please explain it again?
Uh, how do I explain this? Like I mean that if we are talking about so the main function of the pollen in as it is being produced, right?
Yeah.
So is
Yeah.
that functionability sort of retained?
No, 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 no. You cannot germinate another plant out of those pollen. No.
So there is when you are so my question is if you are say retrieving a bunch of samples from an area,
Yeah.
right? So basically those were locked in fossils in the rocks and all that, right?
Yeah.
So is it conceivable that when you are excavating from that area some other pollen grains are also uncovered and at least they're there, so they are now no longer like sort of they're present in openly, so can there be a possible chance of cross pollination from that in a way?
Might
If
be. We
Not
haven't
cross
come.
species, not definitely not that, but
Yeah. Yeah. Actually, it does happen. We do find sometimes transition kind of pollen, we call them, like some characters of one family and some characters of another family are found in a single pollen grain. So, but it has not been published effectively, so we cannot comment very definitely on this. It is just our personal observation in lab.
okay, okay,
Yeah.
thank you.
I think he also
I
means
have a
to ask.
question. Sorry,
Yes,
I'm
yes.
sorry. You can go.
Just a second. Yeah, I think if I understand his question, he's also trying to ask whether the fossilized pollens that you have now unearthed because of your field work
Mm-hmm.
are they able, is it possible that they'll reproduce with the living pollen?
No,
Sorry, what living flowers in the real world right now?
no, no, no. They are not anymore viable.
Yeah.
No.
Yeah, Surbi, you can go ahead.
Yeah, it was really informative. I enjoyed it.
Thanks.
Thanks for this. And I'm no way connected to the field, but I was really curious how you gain access to the mine sites and what kind of collaborations you have with the authorities.
Yeah,
Thanks.
we do write emails to authorities, and then some paperwork gets done, and then we get access to the field.
Thanks.
Okay. Do we have more questions?
Mahi,
Yeah.
something just came to my mind. So,
Yeah.
have you found any pollen grains to which you can relate plants, which you think are of a vegetation, and you can't imagine that vegetation could be in that place in India? Like something that surprised you?
Yes, yes, it did happen to us actually. So, not in India, but like, okay. So, we received some crotinoidiforms in which are not presently which are not presently found in India, but they are distributed in South America or Africa, and those crotinoidiforms are associated with swamps. So, but that type of ecosystem we do not find in India. So we do have Myristica swamps in Western Ghats, but then again, crotinoidy is not associated with that. So with this information, we suggest that at around some few million years ago, there were swamps in India that were associated with crotinoidy and all other assemblages. So that sort of things happen.
Wow.
Yeah.
Thanks.
We have a question in chat box. Considering that the distribution of SAL has already shrunk, what are the future prospects of the work? Does this work also contribute in conservation? It does. It does actually. So for that, we require to do species distribution modeling in future, and that I am still learning on how to do it. Then, with the help of the phylogenetic analysis that I showed in the last, we kind of know that how a particular species contribute to the evolutionary history of its family. And by having that estimate, we know that which plant needs to be conserved or which plant needs to be given more priority when it comes to the conservation policies. Yeah. Did I answer you, Harshada? No, Anusha. No,
Sorry,
Dhanusha.
Dhanusha. Yeah, it's okay. It's okay. No, I didn't have that proper internet, so I couldn't ask you the question like that.
Yeah.
But then, yeah, I was looking like we are putting so much efforts in this study. Like you have put so much efforts in the study. So what are the like future prospects of conservation? And I think you've answered it. Uh, I mean, there are a lot of future prospects, and like you said, distribution modeling. So
Yeah.
then, there is a chance that after 100 years, maybe Sal will be only found in some pockets of Madhya Pradesh.
Yeah,
So,
there are chances, yeah.
so when we can we like can we like keep this pollen grains and then maybe after 100 years, like what is the implementation sort of? Is
No,
there any
actually,
chance that
that
uh,
kind of technology, I don't think has been anyone has come up with that kind of technology that we can preserve these pollen grains and then use them later on to fertilization, for
no,
fertilization.
for example, like we have seed banks. So then the seed
Hmm.
banks are the only way that we conserve this trees for future, or then
I
what
guess
else can
yes.
you suggest?
Yeah, I'm not aware of if this pollen grain can also be preserved because I haven't heard of anything like that yet.
Because I mean, this will be very interesting
Yeah, yeah. If someone would
for
like to work
future
on
and
this,
conservation. Yeah.
yeah.
So thank you so much, and it was a very great presentation.
Thank you. We have Mohit. Considering the size of mine, what sort of characteristics do you look for before digging? So it must have lignite in it, obviously, because lignite is the main source of pollen. Then, if we talk about sandstone, sandstone is basically made up of sand. We do not find any preservation there because of the oxidation processes and all. So, the lithology in which oxidation can occur that destroys pollen grains. So yeah, we do look for the lithology first. It should have lignites, it should have some silts and clays. Then we go for further investigation in the mine. Please give me a second, just let me just put on the switch for my uh, this laptop. Yeah, I'm really sorry for that inconvenience. So did I answer you, Mohit? Hello? Yeah. Well,
I think Arvind has a question. He has raised his hand.
yes.
Yeah, hello. Hi.
Hi, Arvind.
The session was very informative. I'm lucky to have attended it. Thank you so much for showing all your work.
Yeah,
It's
thank you.
inspiring. My question is kind of a follow-up question to the conversation you had with Harshada.
Yeah.
So, so in the present trend, every effort is made to make conservation efforts work.
Yeah.
So, right now we have collected a lot of fossil samples. And all those fossil samples are, do they have these genetic information in them, or the genetic information is also destroyed? I
No,
know they would have lost their viability to vegetative, to their vegetative zone, but do they have their genetic data also? You know,
not from the older time scale. So this
okay,
concept of ancient DNA extraction has been popularized recently, but up to few million years ago, maybe, or I should say up to 1,000 million years ago, that people have been capable of extracting the DNA information from the pollen fossils that are around 10,000 years ago, but not older than that.
okay.
Yeah.
Because if that is possible, even if this collected sample has lost its viability, you can still
Mm.
try to regenerate the
Yeah.
uh, whole code that will actually work, and you can make
Yeah.
conservation possible. Yeah, that was my
Yes.
question. Thank you so much.
Thank you. Yeah. Do we have more questions? Yeah, we have Divya. Oh, just give me 1 more second. I guess my charging point didn't work. I'm so sorry. Yeah, it's all right, I guess. I'm really sorry for the inconvenience again. Oh, we have question from Divya. Regarding the conservation question, I think pollen banks do exist which are preserved cryogenically. All right. So, can you just put some more light on this, Divya? Okay, how much older pollen banks are those? Or maybe these are from the current time period, I'm guessing. Do we have Divya online?
About it. I just like Googled it and it came up about the pollen banks.
Yeah,
Like
but
they are saved using liquid nitrogen and stuff.
yeah, but from the present time period, I'm guessing, not from the older time scales. These are not pollen
Okay, okay.
fossils. Yeah, these are the living
Okay.
ones.
I think that
Okay, I
still
see.
helps Dhanusha's question in some way, that the present species diversity that we have, that is the distribution range of which is becoming smaller and smaller, can still be preserved if pollen can be preserved, right?
Yeah.
Dhanusha?
Yeah, exactly. The same thing.
So if anybody else doesn't have any question, I have one.
Sure.
So I'm curious about what was the diversity richness, the species richness like in those times compared to what it is now, if you have looked at anything like that.
Mm, so when like before identifying the or before claiming that a pollen was present at that time, we do look for its species richness. So when we have received or when we have recovered around 300 pollen grains of a particular type, only then we say that yeah, that pollen was present in that area. So if you compare the past and the present species richness or diversity, uh, we cannot very confidently comment on that because there is a lot of pollen fossil works that still has to be done. Like I have received 8 fossil pollen types of Dipterocarpaceae from Indian sediments, but if we look out for more sediments and for more Dipterocarpaceae fossils, so I might find some other species as well. So yeah, that comparison is really hard to make. But yes, with time, the diversification has increased. This is for sure.
And I'm also assuming that obviously there were a few families that were not present at that time but are present now, but
Yeah,
or and the other way also, that they were not, they were present then and are not present now.
yes, because of the changing climatic conditions and all, like there were some families which were able to survive on the Indian plate when it was situated around the equator, because at that time it used to receive a lot of rainfall. But as it entered and collided with the Southeast Asia, the most of aridity transgressed over the subcontinent and it wiped out most of the forms. So that also happens.
Okay. And I wanted to know if there are any similar studies in Africa, or I think you said America, that some of the species were found in, where
Yes.
they have found species that are, say, now found in the Southeast Asia or the Asia regions in general.
Yeah, there are many studies like that. So from Africa, we do not have much study with respect to the pollen fossils, but in South America, yes, there are several pollen grains which are found there and not anywhere else, or some pollen grains which were found there in the past time, but not now, and now they are preserved or present in Southeast Asia. There are many cases like that.
That's interesting. Thank you, Mahi. Anybody else? Any questions?
No more questions, Mahi. Just best of luck for your future endeavors. Very
Thank you.
interesting.
Just one
Um,
point I want to make here. Pollen itself is a plural word. It is pollen grains if you want to use it in plural sense, or otherwise use pollen for plural as well as singular noun. It's the same. Pollens is not a word.
Thank you.
Yeah.
So one last quick question, if yeah.
Yeah.
So what I wanted to know is, okay, so these are also underground habitats, basically mines. One you're talking about is an open cast mine, but then
Yeah.
whatever you found it is basically from underground, so that it comes in the subterranean category. So are there any studies, very similar studies which people have done in caves or any similar habitats, like if you know in India or
Not
somewhere outside?
I'm no, not I'm aware of. I'm just aware of the mines or the coal deposits. And yes, the
Uh,
Deccan, the coal deposits and the Deccan intertrapians as well. But yeah, not.
But then,
I'm
what
not.
is your opinion? Can we get such kind of
Yes, yes,
fossils or pollen grain things inside caves also? Is it
yes. It is. It is very much possible. But again, that will be of very recent time when we talk in geological terms. Like that depends on the age of the cave. So yeah, and that obviously pollen fossil can decide on it. That what is the age of the cave, and that should be very
okay?
recent,
Right.
I guess. Yeah.
Mm-hmm. Cool. Thank you.
Any closed mines, like underground tunnel type of mines from which these kind of fossils have been unearthed?
Yes, yes. And in Meghalaya, there were closed mines actually, where not me, but my guide and all have explored those mines and taken out samples from them.
What kind of mines are they? Like, so what is the mineral?
Coal deposits. They were coal deposits
Okay.
actually. Yeah.
Oh, nothing from limestone or sandstone area, like yet.
No, no, not from sandstone.
Not
As I
suitable
just
for.
mentioned, sandstone, yeah, that cannot
Mhm.
preserve pollen grains because of the oxidation process.
Okay, so both the rocks, not even limestone, not even sandstone, they cannot. They don't
Yeah,
have that
they can.
properties.
No, no, they cannot
Okay.
sustain.
Oh,
Yeah.
this is interesting. So, what are the rock types which can actually sustain
Silt,
pollen grains?
silt, and yeah, silty shales, silt, and lignite coals. All these forms.
Oh,
Okay,
shale.
thank
Shale
you.
also that.
This
Shales.
is
Yeah,
very interesting.
yeah. I guess you guys might find shales in caves, right?
Yeah, yeah.
Yeah,
Yeah.
yeah. We have shale caves in India.
Yes. So you can get fossils from them, pollen fossils from them.
Yeah, I think
Right.
Arvind has a question again.
Yes. Okay. So this whole topic is Greek and Latin to me. So my question may sound very elementary, but
That's okay.
I still want to ask it off. So does the type of mine affect the kind of samples you collect? Say,
Oh,
for example, I know mine is basically a place which is formed millions and millions of years later after something has happened. But does the type of work that's happening there, or what mineral they're extracting from that point, affects the samples you extract from that place by any chance?
no, I don't think that affects the presence of a uh, pollen there. So I don't think it really does matter that what kind of wine it is and what kind of flora we can extract from them.
Okay, because somewhere down the line, they're going to bombard the whole area. They're going to extract minerals from there. So,
Hmm.
don't you think there'll be a chance for all these preserved samples to get damaged? Before we collect, it's not a sample; it's just part of the mine area.
Yeah, the pollen fossils might get broken, and like I just, they cannot be destroyed completely, but yeah, part of it will still be preserved. But yes, again, you are right that based on that part of the fossil, you cannot identify it to family level or the genus or species level. That
The
is the case.
second, the second fossil image where only some tooth were seen, uh, like that.
Sorry, I
In
didn't
your
get you.
presentation, you were showing a few pictures. There was a
Huh?
mammal and there was another animal, which was unidentified. Only a part of that animal was fossilized.
Yeah, that was because I do not work on animal kingdom. That's why I couldn't identify it. It's not like it is not being identified.
Oh, okay. So
Yeah,
bits and pieces of the fossil, when extracted, can still be identified.
yeah. So if
Oh,
you have certain length of trace fossil, you can still identify it.
okay. And I'm so sorry to take your time, but how? What is this method like? How do you do it? Like, how do people do it? You?
What the identification part, or
Yes, yes, identification.
yeah, like I mentioned, we have to refer a lot of literature or a lot of work that is that is related to the morphology of the creatures in the present time. And then, yeah, by the comparative analysis, we can very definitely assign the living counterparts to our fossils.
Okay, okay. Yeah. Thank you.
I had a
And
small question.
yes, Shraman.
So, say you want to start this kind of a work. Say I want to start this kind of a work. So,
Hmm.
how do you go about it? Like, what's the first step? How do you pick a site and then go about with the collection process?
Oh, refer to the literature. See, so from the geological sites from where people have already did some pollen extraction work, and then look for the lithology basically at different sites, and then go for it. Like if you think that this is a particular site from where people haven't much extracted pollen from, so you must go for that site. So in my case, people like most of the work was already being done in case of Bastan lignite mine in terms that they have identified the fossils, but they had not reconstructed its vegetation and paleoclimate. So that's what we decided to go for.
Okay, thank you so much.
Yeah.
I have one question. I'm not from this field, but I'm just curious since I'm from Meghalaya and we have like
Now,
a number of fossils like in Meghalaya. So
yeah.
like basically you study the like when there is an imprint. Let's say like recently like we found this by my assistant center like a photo where there's this imprint of a leaf. There's
Okay.
a clear impression on the like probably like I don't know which stone it is exactly since I'm not
Hmm.
from the field though I study plants. So like in that case, like you first study the area and the stone as well. I mean like the object where the imprint is found. Is it something like that?
Yes. You have to study the lithology as well because that also indicates some sort of environment in which the fossil was deposited. So that is done by sedimentary geologists.
Okay, okay,
Each
okay.
kind of lithology represent a different kind of climate. Yes.
Okay, then based on that, like the type of the sedimentary rock or that you will like be like you'll carry out your like study.
Yeah.
Okay, okay. Thank you so much.
Thank you.
Mahi, this reminds me, is it possible to identify the family or something like that from a leaf fossil?
Yes, it is possible. Yeah, based on the different characters of leaf, people do identify the leaf fossils up to species level.
Oh, wow. Okay.
I have one more question then,
Yes.
since since that fossil like we just like saw like a week ago.
Hmm.
So like in that case, you said like we can like identify at least to the family level. We are also guessing a certain family in the lab,
Okay.
like among like seeing just a morphological character. So
Hmm.
like when you said like you can like identify up to like the
Species
family,
level. Yeah.
yes, or the like the genus level and all. So like do you also just identify looking just at the morphological character or you go like into like further kind of like study?
No, just based on the
I mean like
morphological characters,
okay,
because
okay, okay,
I don't think any other sort of information can be extracted out of these fossils that are too old. Yeah.
okay, okay, okay. Thank you so much.
Do we have more questions?
So jumping on to what the thing we were discussing. So
Hmm.
sometimes it can be like that uh, the same species in different places, they
Yeah.
can end up showing similar or very different characteristics, right? So if we talk
Yes.
about phenotypic plasticity, say,
Yeah.
so how do you deal with that if you're looking at a fossil and identifying it by morphological characteristics only and not going into genetics? Wouldn't that be a problem?
Not really. So, no, not really. So we haven't encountered this kind of problem yet. So if there is a phenotypic plasticity that lasts for a certain period of time, actually. So say if you are talking about Paleocene, Paleocene is around 63 million years ago. So a particular species will have similar kind of morphology over that period of time, and then when we go further in time, towards the recent time, actually around 50 million years ago, that is Eocene. So the morphology changes, and so this morphology changes in all these species that are spread across the globe. Yeah, that's how this happens. If phenotypic plasticity is there, then it does spans to a time interval.
Okay.
Is it not possible to extract DNA from such kind of fossils? Say leaf fossils especially.
As I said, so this ancient DNA extraction method is applicable to recent time period in terms of geology, not from much older time scale. So it is possible to extract DNA from these leaf fossils, but up to few thousands of years ago,
Okay.
not in terms of million years.
So anybody else, any questions? The feedback link is in the chat box. If you would like to leave your feedback, anybody we'll especially like to hear from people who are not from the field, and how did you find the talk? Because I think it was an excellent, very simple and clear talk. So I think that's it for the questions. We can, I'll just
If you have more
say my
questions, you
thanks to
can
Mahi.
anyway, email me. If you have more questions, you can anytime email me.
Yeah, I
Mahi,
think we
can
can
you
drop
just
your
add your
I'll
email
just
ID? Yeah, that.
yeah. So I have just dropped my email ID in the chat box. So you can anytime drop me a message there.
That's great. So thank you so much, Mahi. This
Thank
was
you, Amrita, for
seriously
having
a
me.
very interesting. Yeah.
Thank you for having me. Yeah,
no, it was very interesting. I think even for us, we are not really, although being from the field, we are not really from the paleology background. So for me also, it was very interesting to see how right from coastal forms to upland flora can be unearthed from such mines. And I think your presentation was really good and very eye-opening even for speleology in itself, because rarely is mines talked about in terms of speleology, and it really shows the basic interdisciplinary like character of this kind of a study, that you have geology and biology, and in biology, then botany, and also speleology. So thank you so much for coming here and talking to us,
it
and
was
for
a
answering
pleasure.
all the questions, and all you participants. Really good questions, really good interaction. Thank you so much. And yeah, that's it. The webinar has been recorded and will be uploaded to the YouTube channel, but we also have like other 4 webinars that we've done in the past. You can also look them up. They're also very interesting talks. And yeah, thank you so much everyone for coming here today. अनुषा.
माहीने पण आय थिंक लीव्ह केली मीटिंग.
हो, तिला सांगू का काही बोलायचं असेल तर सांगते, नाही तर राहू दे.
हो राहू दे मग. अच्छा मॅम आले. हाय मॅम.
Hi.
Yeah. Good.
I'll
One
just
second.
tell
One
her
second.
to say.
1 second. I'll just stop recording. नाही तर मग ते परत.
Yeah, yeah.